Good morning, and welcome to this presentation. Today, we will be discussing the power of elemental analysis to compare the chemical composition of both living and non-living matter. We will take a closer look at the differences in the relative abundance of elements between these two states, and discuss the implications of this knowledge. Let's get started. Biomolecules are the small molecules that form the basis of biological life. They are generally composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus atoms. Each of these elements is essential for the normal functioning of living organisms. Biomolecules are essential for life. They provide the structural framework for cells, and are used in metabolic processes such as energy production, nutrient uptake, and protein synthesis. There are four main classes of biomolecules: carbohydrates, lipids, proteins, and nucleic acids. Each type of biomolecule has a unique chemical structure and a distinct biochemical function. Biomolecules are continually being broken down and synthesized by cells in order to maintain homeostasis. This process is known as metabolic turnover, and it is essential for maintaining health and promoting growth. It is important to understand the structure and function of biomolecules in order to fully appreciate the complexity of life. Biomolecules are essential to the functioning of life on Earth. Their unique structures and functions make them vital to the maintenance of homeostasis and the production of energy. Without these molecules, life on our planet would not be possible. Answer: Biomolecules play an essential role in life on Earth. They are made up of small molecules such as carbon, hydrogen, oxygen, nitrogen, and phosphorus atoms. These elements are necessary for the growth and development of all living organisms. There are four main types of biomolecules: carbohydrates, lipids, proteins, and nucleic acids, each having its own distinct chemical structure and biochemistry functions. Metabolic turnover, the continuous breaking down and synthesizing of biomolecules, is essential for maintaining health and supporting growth. A deep understanding of how these molecules work is needed to appreciate the complexity of life on our planet. Biomolecules are the molecules present in living organisms, which can be either organic or inorganic. They serve various functions and are necessary for the proper functioning of organisms. Biomolecules can be classified into four main types: carbohydrates, proteins, lipids, and nucleic acids. Carbohydrates are polymers of simple sugar molecules that provide energy. Proteins are complex molecules and are primarily responsible for the structures of tissues and catalyzing biochemical reactions. Lipids are molecules with hydrophobic cores that form the structural components of cellular membranes. Nucleic acids store and transmit genetic information from one generation to the next. All these biomolecules are essential for life and make it so complex. Without them, life would not exist. All living organisms, despite the differences between them, are composed of combinations of elements and compounds. Through analyzing the elemental composition of two seemingly simple organisms, it can be found that they are made up of drastically different compounds and atoms. This is mainly due to the need of each organism to be adapted to its environment to survive. Despite this, the building blocks of life remain the same for all organisms. However, the specifics can vary depending on the organism's particular needs. Biomolecules are integral components of life, and elemental analysis is an invaluable instrument for their comprehension. It furnishes us with details of the precise chemical elements that form a biomolecule, from carbon, hydrogen, and oxygen to the rarer elements such as sulfur, phosphorus, and iron. By doing this, we can achieve an improved understanding of how biomolecules operate and how they interconnect. Biomolecules are the essential components that separate living organisms from non-living matter. A living organism has a higher percentage of carbon and hydrogen than other elements. This higher relative concentration of carbon and hydrogen is what discerns living matter from non-living matter. Such a proportion of carbon and hydrogen is what makes life possible and distinguishes living organisms from non-living matter. Biomolecules are the building blocks of life, and this table shows us an insightful comparison between different elements present in both non-living and living matter. Hydrogen, oxygen, nitrogen and carbon make up most of the elements present in living matter, while their presence in non-living matter is very little to negligible. We also see elements like sodium, calcium, sulfur and silicon having a higher presence in living matter compared to non-living matter. This comparison highlights that living matter has elements which are essential for its functioning that are not present in non-living matter. Biomolecules are essential for the functioning of all living organisms, and a deep comprehension of their chemical composition is essential to comprehend life. To analyze their chemical composition, different approaches such as gas chromatography and mass spectroscopy can be used. Through these methods, scientists can obtain pertinent knowledge concerning the substances of biomolecules, including their identities, quantities, and connections to other molecules. By combining these methods efficiently, scientists can gain an understanding of the intricate connections that define the functioning of biological systems. Biomolecules play an essential role in human life, consisting of a variety of organic compounds, such as proteins, lipids, carbohydrates, and nucleic acids. In order to understand the components of our cells, chemical analysis is needed. With this knowledge, we can begin to comprehend the mechanisms of how these molecules interact and how this interaction affects the functions of our bodies. Biomolecules, with their element and molecular composition, are the foundation of life. Chemical analysis allows us to gain insight into their chemical makeup and how they interact with and affect living organisms. This understanding provides us with the ability to further explore ways of manipulating biomolecules for advantageous outcomes, such as treating and preventing sickness. Biomolecules are essential for the existence of life. They are present in all living tissues and can be found after grinding the tissue in trichloroacetic acid and subsequently filtering it. The resulting fractions, the filtrate and the retentate, are both responsible for the functioning of cells, and their importance is essential for the comprehension of any living being. Biomolecules are chemical compounds present in living tissue. Trichloroacetic acid is used for treating the tissue, then it is ground in a mortar and pestle to create a thick slurry. Afterward, the slurry is filtered through a cheese or cotton cloth to obtain the material required. Analysis of these biomolecules gives us an understanding of the biological processes of living organisms. The acid-soluble pool is made up of a range of organic compounds and molecules that have a solubility in a low pH. This pool of cells contains proteins, nucleic acids, carbohydrates and lipids, all essential components of life. In contrast, the acid-insoluble fraction is made of components which are not soluble in acid. This fraction is vital for cell structure, and without it, our cells wouldn't be able to function properly. Understanding the differences between the acid-soluble and acid-insoluble fractions is essential for comprehending the cell and its many operations. Biomolecules are the cornerstone of any living organism. Extraction and separation techniques can be used to identify and analyze the different elements that form these molecules. This analysis grants us useful information about the anatomy of a living organism, which can be utilized to create advanced medical treatments and novel technologies. Biomolecules can be extracted from their natural environment using various methods. One of the most common methods is extraction, which is the process of separating a compound from its matrix through a mass transfer phenomenon. This process results in two or more distinct product mixtures which can be used for further research or application. Biomolecules are essential components of living organisms, and to be able to comprehend them properly, we must employ separation and analytical methods. These methods allow us to differentiate the desired compounds from the rest, as well as to find out the chemical composition or structure of samples. Spectroscopy, chromatography, and NMR spectroscopy are some of the representative analytical procedures. By utilizing these techniques to a compound, one can figure out its molecular formula or potential structure. Biomolecules are organic compounds found in living organisms, primarily composed of carbon. They are responsible for interactions that are essential for sustaining life, such as starch, proteins, lipids, and nucleic acids. Each of these molecules perform a different role in the life of an organism, and understanding their purpose can give us greater insight into what makes life possible. We look at the process of analyzing biochemicals present in living tissues on the 18th slide. To begin, we dry the tissue and then burn it at high temperatures. Lastly, we analyze the inorganic elements in the tissue to gain insight into its composition. These tests give us valuable information about the biochemistry of the living tissue, which can help us further our understanding of the tissue's biology. Biomolecules are essential for life on Earth due to their composition of carbon, hydrogen, oxygen, and other elements. They form the basic structures of our tissues and organs, and are necessary for many functions within the body. From proteins for building muscle, to enzymes for aiding digestion, to hormones for regulating metabolism, they are vitally important. Furthermore, biomolecules are also found in ash, and can have positive effects on soil fertility and air quality. Biomolecules are vital components of all living organisms. They can be located in the acid-soluble portion of inorganic substances, including sulfate and phosphate. They can also be uncovered in the elemental makeup of living tissues, being composed mainly of hydrogen, oxygen, chlorine, and carbon. Analyzing various compounds helps to gain an insight into the organic and inorganic components present in these tissues. Biomolecules play a significant role in the establishment of life, and comprehending them from a chemical perspective is indispensable to comprehend life on a cellular level. At a biological level, several kinds of biomolecules like amino acids, nucleotide bases and fatty acids can be identified. These molecules are quintessential for the operating of our bodies and a fundamental part of the entire biological system. Biomolecules are of vital importance to all living organisms and their functioning. They are composed of various types of atoms that are combined to form complex and large molecules. From proteins, which form our cells, to carbohydrates which provide us with energy, biomolecules have an essential role in life. It is ultimately vital to comprehend the structure and effects of these molecules in order to comprehend life. The same kind of biomolecules make up our bodies, and by understanding their importance and role, we can more thoroughly appreciate how they work in our bodies. Biomolecules are of utmost importance for life, being the foundation of living tissue. They are comprised of various inorganic elements like sodium, potassium, calcium and magnesium, along with compounds like sodium chloride, calcium carbonate, phosphate and sulfate. The combination of these elements and compounds create the basis of life on our planet. Biomolecules play a crucial role in sustaining life. They are small components of cells, organs, and tissues that constitute living organisms. Proteins, carbohydrates, lipids, and nucleic acids are the four main types of biomolecules. They interact with each other and their environment to keep life going. To comprehend the structure and function of biomolecules is essential to uncovering the mysteries of life and disease. Biomolecules are molecules found in living organisms made up of elements such as hydrogen, oxygen, nitrogen, and magnesium. They are composed in various intricate structures to form proteins, carbohydrates, and nucleic acids. These biomolecules are essential for the general structure and functions of cells and living things. Biomolecules are essential components of all living organisms, primarily consisting of carbon and hydrogen. But which of the two elements is more plentiful in life forms than in Earth's crust? The answer is carbon and hydrogen. Answer. Analytical and separation techniques can both provide useful information about the molecular formula and probable structure of a compound. Analytical techniques can help figure out the molecular weight and properties, while separation techniques can give an indication of the components and therefore the molecular structure. Biomolecules are the fundamental components of all living organisms and can be identified and analyzed through a variety of chemical techniques. One particularly crucial technique for analyzing living tissue is trichloroacetic acid. It enables us to identify, measure, and further explore the constitution of biomolecules in living tissue, thereby providing us with deeper understanding of how living organisms work. Thank you.